if anyone needs to recap anything later on for you guys as well. Okay, so we'll take down that poll now and we'll get started. So let's just show around Collaborate first. The first thing at the very bottom of your screen, you'll see a little picture, and it's a bit like a cutout picture of a human. That there allows you to do emotions, exit the session, tell us to speed up, slow down. So what I want you to do first of all is click on here and click one of your emotions, tell me to speed up, slow down, agree, disagree. So just quickly do that so you guys know where it all is. How are we going there?
Yep, got heaps of faces.
Awesome. Hopefully we're all happy today. Now the next one is your mic. If you want to answer a question, you can do this. The last one, or the next one, sorry, is your camera. You can put it up, but we just ask that you don't have all the cameras up because it slows down our feed. So we just request that only having like one camera on who is the presenter, it just makes it a bit easier. The last thing is your hands up. So could you all raise your hand now, just so we know that you know exactly where to go? Perfect. Okay, and now the next step is to lower your hand. So just clicking straight on there again. So lower it just so we know that you guys are all over it. The last one is at the very bottom here. You click on this purple arrow and it will pop open a panel. And in that panel, it has a few different spots that you can go in. The first one has like a speech bubble. Click on there and we want you to go into the everyone chat and just write hi or I'm having a great day or something like that, just so we know that you know how to communicate with us if you're having any issues later. Perfect. Awesome work, guys. So we're going to chuck up a poll question in just a minute. The first thing is we've got these three images. We've got bacteria, we've got a frog that seems to be glowing, and we've got insulin. The big question is, how do these all link together? So we'll get a poll question coming up. Oh, sorry, in the chat, sorry. In the chat, my apologies. So in the chat, you'll see a question and see if you can answer it. Your bit of a clue here is your pre-work. If anyone's having any problems with hearing us, just message Etienne or Mabel and they'll be able to help you out. The other thing is if your internet glitches at any point, you can jump into your Canvas and most of the instructions are in there as well. Otherwise, we're recording this session, so if anything happens, you can watch later. So let's have a look at those answers. What are we putting our link as? And it's okay, guys, if you don't know, because hopefully by the end of this session, we'll be able to have a bit of a chat about it. Gene manipulation, nice. What else we got there, Alex? Modification, manipulation of genes. I like where you guys are heading. Awesome. So we're now going to jump into our next slide. And this is basically a bit of a cap or an outline of what we're doing today. So we've already done our introduction to navigating online. We're then going to look at the story of insulin, so where it all began. We're then going to have some pipetting skills, and you guys are going to do this virtually, it's just really cool, and I'm going to do it live here. Then we've got our bacterial transformation, and we're going to do this together. So I'll do it in the lab, and then you guys will do it in the virtual lab. And then we're going to explain what the GFP gene is. And then we're going to predict our growth of our plates. But before we do that, we need to give you a bit of terminology of what the growth of the plates will look like. And then we're going to have fun streaking the plates in here, and you guys are going to streak them on your virtual lab. Then we're going to look at our observations and analyse those plates. The last thing is our plasma digestion. And if we have time to do it with you guys, we'll do it virtually, but we'll be doing it in here live. It's just depending on time, and then we'll run our products in a gel. And it's a bit of a, here's one we prepared earlier because it takes 45 minutes for the gel to run. And then we're going to have our conclusion. Now, just a bit of a housekeeping as well. You need to have your Canvas open as well as Collaborate. So can everyone raise your hand if you've got your Canvas open on another tab? If you haven't done so already, please do so now. Or if you're having any issues, just let us know. So we definitely need both your Canvas and your Collaborate, which is where we are right now, open. Has everyone got their hands up?
There's people lowering their hands.
So can we just leave our hand up if we've got Canvas open so we can double check?
It looks like the vast majority.
Okay. So if anyone's having any issues with Canvas at all, with your log-on or anything, just let Etienne or Mabel know. The other thing is you're going to be opening up other tabs as we require. So your virtual pipetting, your WOW, BioLab, and then the restriction mapper. Now the restriction mapper depends if we have time. All the instructions are on Canvas, so you can do it at a later date. It's okay. And as for all us teachers, we love our learning intentions. So hopefully by the end of this session, you'll be able to determine and analyse the results from a restriction enzyme digest. You'll be able to observe and simulate in a virtual lab the techniques and understand the process of bacterial transformation. And the last one is to evaluate that particular bacterial growth and determine which bacteria have actually been transformed. So we're just going to watch a short little video, if it will play for me. There we go. And hopefully, fingers crossed, we've got sound.
Insulin was introduced to the world in 1922 at the University of Toronto. The scientists went to a hospital ward full of diabetic children, most of them comatose and dying from diabetic ketoacidosis. This is known as one of medicine's most incredible moments. Imagine a room full of parents sitting at the bedside, waiting for the inevitable death of their child. The scientists went from bed to bed and injected the children with the new purified extract, insulin. As they began to inject the last comatose child, the first child injected began to awaken. Then, one by one, all the children awoke from their diabetic comas. A room of death and gloom became a place of joy and hope. Thanks, Dr. Banting, Dr. Beth.
Okay, guys, so that's your quick story of insulin. And basically with insulin, it's for people who have diabetes. Does anyone in the room know anyone that has diabetes, whether it be type 1 or type 2? Yeah? It's a lot of people will know someone that has it. And it's a bit of a scary thing because back in 1922, before then, there was nothing they could do. So if someone had diabetes, they pretty much ended up dying, which is really sad. So when they found that they could take it from a pig or a cow, they then were able to inject it into the people and they were actually recovering, which was amazing. The last thing, which was the biggest step, which was in the 80s, was when they were able to grow it up themselves. And this is where this technique starts to come in, which is pretty amazing in itself. So the first thing, we're going to go over our first prac. So the aim is to transfer bacteria, or transform, sorry, and determine the results of the bacterial growth. Now, all your materials and methods are up on Canvas. So if you were with us, you would actually be doing this prac. So it's a bit sad because we would love for you to be here doing it, but sadly during the times we're not allowed to. So now I'm going to stop sharing my screen and I'm going to show you how to use a micropipette. But before I do that, who here has used a micropipette before? Just raise your hand. How many of you have had the amazing ability of using a micropipette? Just raise your hand. Looks like a few of you have. Perfect. So I'm going to show you this one here. And this one is a 0.5 to 10 microliter. And the first thing you need to find is this knob at the top here. And you've got to find what is called the first stop, which is here. And that pushes out all the air out of the barrel. And that allows you, once you need to go and collect your liquid, to draw it all up. Once you've got your liquid, you then put it into the container that you want to inject in, and you push it all the way down to the bottom, which is your second stop. The other important thing is setting on the front here. You need to set its value. So you simply turn this around, and as I was saying before, you need to be really conscious of what or how much volume you can put in here. So this one can do from 0.5 to 10 microliters. So you simply turn that and it tells you what value. So at the moment I'm at 9.2. So I'm going to grab a pipette tip, and you always want to make sure that you close your pipette box afterwards because you don't want any dust or any contaminants in there. So if I was to get a liquid, I would simply go to it, first stop, go into my liquid, draw it up, and then go to my next container that I want to remove or put it into, and push it all the way down to the bottom, and that's where it would go into. And to remove it, you simply push this button here on the outside to eject. So instead of me standing around showing you how I can eject tips and all that fun stuff, I'm going to jump into the lab now, and we are going to run through this together. So I'll share my screen in just one moment. And if you don't have your Flash Play enabled, it won't work. So I just want to show you how to do this because it's a little bit confusing otherwise. So bear with me just one moment. Okay. So is everyone now seeing the screen? Perfect. So simply, I'm just going to show you and then we'll get you guys to go on first because there's a few little things you need to learn about. So we're just going to do level 1, and you guys can go back and use this at any stage. So it's really important. It tells you how to add a tip, tells you that you need to set your volume, draw up your liquid, and that's what I was just talking about, that first stop and expelling. And you've got your red dye and all your different pipetting equipment. So your P2 needs to use the P2 tip box and so on. And the last one, which we all love as teachers, is having predictions. So you need to go through and go, oh yes, I think this one's going to be 20 microliters. Oh yes, this is going to be 15. Oh, that looks a bit too big, and so on and so on. So then once you've done that, you then start, and you need to read what the instructions are on the side. So it says use your P20, so you collect your P20. The first thing it wants you to do is set your volume. So you click on the side here, and you simply push your plus or your minus to get your correct volume. When you're happy, you press save, and then you can go get open up your box. So it gives you lots of hints along the way. You collect your tip, okay, and it tells you to close because we don't want to have any contamination. You then come down and get your red dye. You put it in there, and now you've got to go to your first stop, draw it up, and then you push it all the way down to the bottom, second stop, and then make sure you eject and close that red dye as well. Eject. When you're in here, eject your tip. Now, if you happen to come and get a tip and come down here, get your red dye and go all the way to the first stop, if it lets me, the second stop, sorry, then it will tell you to eject the tip. So if you do the wrong stop, it will ask you to move. Okay, so I'm going to give you guys 7 minutes to try and get 2 of the micropipetting done. If you do more, fantastic. If you don't, that's okay. And when you get back, there'll be a question waiting for you guys.
Yeah, sorry guys, we forgot to put that micropipette link in the chat earlier. So if you're completely lost, Eddie's just popped that in the chat for you. So click on that and you'll get to what that was in before.
All right, guys, we'll just give you 5 more minutes, and when you're back, there'll be a poll question waiting for you. So 5 more minutes, and there'll be a poll question waiting for you. So try and get at least 2 of the micropipetting done. All right, guys, 2 more minutes. If you're not already back, start heading back to collaborate. Okay, everyone, if you want to come back into Collaborate now, and there is a poll question waiting for you. If you haven't answered it, please do so. So we're testing a bit of your knowledge now. Yes, you do have to do some maths when it comes to the wonderful world of biology. Okay, showing the responses now. We've got 2 more to come. Who are we waiting on? How's his connection? Everyone's on? Awesome. Okay, so most of you are all over it, so well done. Okay, we'll remove that poll now. Now, the next thing is we're going to jump into showing you the start of how we actually start to transform our bacteria. So I'm just going to show you my equipment first. I'm just going to wait for Alex so he can move around. So really important because we're going to be doing aseptic techniques. So we're going to need a Bunsen burner. Now, any of you who've done chemistry before will probably remember this from the flame test. This is your inoculation loop. So it's a little loop at the top here. And because it's a nichrome wire, you can actually heat that. Now, some kits, you actually have plastic ones, and these plastic ones cannot actually be put into the flame because they will melt, but they are sterile. So it depends on which kit it is as to what you can do. So I'll just put my hand behind it so you can see the little loop there. It's a tiny little loop. We also have our other equipment, such as our micropipettes up here. We have our plates because we need to have them pre-cooled. It makes it easier. We have a few variety, I should say, with our micropipettes. So we have our ones which do larger volumes all the way to our smaller volumes. We also have our different pipette tips, and then we have what we're starting with, our epidorphin tubes. So we have some that aren't labelled, and we have some with our transformation liquid and our pea glow. So the only other thing is we've got our little floater here. It's really important. It sounds pretty funny. And we're going to be putting our tubes in here and then putting them into the water bath. So let's get started. The first thing we need to always do is label everything. So we are going to be doing a plus and a minus pea glow. Who can tell me why I have a plus and a minus P glow? Can anyone tell me? And you also want to write it on the side just in case we rub it off. Who can tell me why we have to have a plus and a minus one? It's a bit of a hard question. I'm being a bit evil with that one. If you don't know, that's fine, guys. Hopefully we'll be able to find out by the end. Cool. That's all right, guys. Okay, so the first thing we're going to do is we're going to get our transformation liquid in, and we need 250. We have an answer.
Someone said P glow is the.
It's the teacher.
Okay.
I'll just give them a context of what you're asking, because a lot of them
No.
they couldn't get into the pre-work.
Yeah, no
Right.
worries.
You just need to kind of have a bit more of a context of what you're talking about.
Thank you for that, Elise. Yeah, so it is our plasmid. So a bit of a clue there then. Why would I have a plus P glow if we're talking about plasmid and minus? See if you can get it from here. You got a big hint right there from your lovely teacher. With the P glow is the recombinant plasmid. No, I think we're all asleep. That's okay. All right, so we're going to get our first lot ready. And I need my bigger tips. Yeah, these ones haven't been opened. So we've set it to 250 microliters, and we're going to get our first one, and we're going to put this into both of our tubes, but we're going to have to change our tip each time. So we get to our first stop again. We then go into our liquid, draw it up. So that's 250 microliters in there. So I'm going to put it into my minus and push it all the way down to the second stop. Close that lid, and then we're going to put it into our waste. And then we're going to do the same again for our plus P glow. So find that first stop again, into the liquid, and then we're going to put it into our plus. Oh, shot some of that out. Doesn't want to eject. There we go. Okay. So the next thing once we're done there is we need to get our lovely inoculation loop, and we need to get our bacteria. E.T., can you grab my bacteria, please? Because it's in the fridge, keeping cold. And we only need to pick up 1 to 2 colonies when we do this. So I'm just going to move this over here so it doesn't melt. We're going to light our Bunsen burner. Oh, doesn't want to light today. There we go. Oh, that just put it out beautifully. On the plate.
On the straight plate, yep.
Yep. There we go. Thank you. Just bear with us one moment. So we've got our safety flame all ready to go. And we're going to be using our working flame. So we're going to turn that around. And hopefully this doesn't go out because it gets a bit too much gas here. There's that nice sound. And all you want to do is heat it there and just get it nice and glowing, and then it will cool down pretty quick. Thank you. Okay, so before I do this, I'm going to redo that again anyway. I just want to show you our colonies here that have been grown. Yep. So you can see how this has been streaked. So you've got your first little lines here, and then you go the next 3 here, the next 3, and the next 3. So later on, we'll be streaking up our own plates as well. So this is just plain E. coli that's been grown. Okay, so we'll get back to our working flame. Put that there for a second. We're going to reheat. Okay. And you want to keep it near the flame because this allows us to not have anything going onto our bacteria. And we're only going to grab just a couple colonies. Do you want to come in? So you only want about at least 1 to 2 colonies. Colonies basically means a little circle. Are you all good? So you just get a little scrape on there. And then you've got to put your lid back on. So I'm going to do this for our Heat Glow Plus first. And you simply just do like a little spin around. Okay, and put it back on. It's a bit hard to see, but there's 2 little colonies just floating around in there. Yeah, flicking it does help. Okay, we'll put that just in there for now and we'll reheat, and we're going to do the same again. So when you go to do the virtual lab, you don't have to use a Bunsen burner because they have it done with a sterile loop. So once again, we're just going to grab 2 little colonies. Go, put that on there. And it's a bit weird because you actually have it upside down, which is a bit freaky. Give it a little stir. There we go. And then you're going to have to burn that again. And we'll leave that off and we'll give it a little flick. And it's just sitting there at the bottom. It's very tiny. Now what we're going to do is I'm going to put that back onto our safety flame. And we're going to put it into our little floater and we're going to come over to our water bath. Thanks, ET. You're right. And we're going to put it into here. It's going to be in our water bath for about 25 minutes. So as we were saying before, we're going to have a bit of a, here's one I prepared earlier as well. So now the next thing is we are going to be jumping into your virtual lab. But before we do, I just want to show you where you're going, and Mabel's going to pop up the link in just one moment, but I just want to show you where you guys need to go first. So I'm just going to share my screen again. So we're going to the WOW Biolab. And when you get into there, you'll see you've got heaps of options, which is really good as a teacher. There's lots more things that you can do with your classes. So we're going to go to your bacterial transformation. Click on there. Now, if anyone has had any problems with their Flash player, when you're in here, it will come up with saying like you need to enable. At the top here, you simply click on there and that's where you can enable things. Sorry. So just at the top there, you'll be able to click on there and if anyone needs any help, just let us know. So when you go in here, you've got all your information. This will actually speak to you, which is nice. You don't actually have to read it all. Then you come in here, you're going to have to go through and click all your different bits of instruments because it won't let you start without clicking them all, which is a little bit annoying, but it's really good because you guys get to learn all the different bits of equipment. So the only thing I haven't done is the incubator. Okay, now you come up to your procedure, so your method, you jump into here and you jump into your wonderful world of predictions. Now before you go on and do your predictions, what we need to do is actually explain a little bit more. So I'm going to jump out of this and we're going to jump back into our PowerPoint because we need to explain what the growth and everything means. Okay. Alrighty, so I'm just going to hide that. Okay, so we've started our lab, we've shown you the equipment. The next thing we want to explain is what do these plates mean? Now, do we have a little poll here, Mabel, to chuck up? Because we need to explain what our plates are doing. So we've got A, B, and C.
So just in the chat, I've just said which plate, A, B, or C, shows a lawn growth. So Josh and Caitlin, great, it is A.
Awesome. Well done, guys.
Great.
So remember I was saying I was collecting colonies. Which one do you think is the colony growth then? Awesome.
Yeah, well done, everybody.
You guys are all over this. And the last one, of course, then will be no growth. Perfect. So let's explain what we were talking about before in regards to our pea glow. So our pea glow is our plasmid. It is a really cool little thing. And it's just a circle piece of DNA. And it has a promoter, and it's called the GFP. Okay, so it's the green fluorescent protein. And as you would have learnt in Unit 3, you would have learnt about making of protein, so your transcription and translation. So you go from your DNA to your RNA, which makes your protein. And this is your protein here that with the presence of a sugar starts to glow. If you don't have that sugar present, and it's gone blank on my mind, which sugar is it again? Arabinose, thank you, I've just gone blank. And when you get sugar present, arabinose, it will then actually make this happen. So this is the promoter. If you don't have the sugar present, it will not happen. Doesn't mean that the actual plasmid hasn't been taken up. It just means it will not be expressing. Now down the other side here is your gene of expression. In our case, if we were talking about insulin, this is what you would be putting in here. So you would be cloning in your bacteria. And then once you've actually got your particular gene being expressed, you then have your promoter, which is your arabinose. Therefore, when you put UV light around, it starts to glow. It is really cool. Very simple tool, and it was, they started, I said, in the 80s. So, green fluorescent protein is your GFP. Does anyone know where it comes from? So you've got a poll question popping up now. Where do you think it comes from? Have a guess if you don't know. Do you think it comes from glow sticks? Do you think it's sea jellies? Do you think it's your dinoflagellates? Or is it just man-made? Have a guess if you're unsure. Okay, let's show those responses. So well done, guys. It comes from a sea jelly. Very cool. So scientists actually use this as a marker. So it gives them a quick snapshot to go, okay, I'll put this on UV. Yes, I can see that this particular bacteria has taken up that plasmid. And the fact that it's glowing means that it's recombinant, which means it's been transformed, which is pretty cool. Here is a really interesting picture. And this is what sometimes it is used for, is to actually see if animals can glow. And as you can see here, if it is in the cell because of cells going through mitosis, you actually will have it going through the whole body. So the whole animal will glow under UV. And of course, you need to feed them the correct sugar, arabinose, for this to happen. The last thing I wanted to talk about is your gene expression. And gene expression as you guys should all know, that you need to have some sort of promoter. In this case, our arabinose sugar is promoting it to actually make this particular process happen and having the protein expressed. The last one is your AMP gene, because you only want the particular bacteria that has the plasmid in it to grow. So therefore, having the AMP gene in it means that if you have ampicillin on your plate, it will kill the others. So your AMP-R allows you to tell if it's got antibiotic resistance to ampicillin. Your ARC is the fact that you know that it's going to, when have the sugar arabinose present, it's going to start producing your protein which allows it to glow, and that protein is your GFP. So as I said before, I've shown you quickly how to get into your virtual lab, but before we do that, I want you to jump into Canvas and I want you to quickly do your prediction. So you've got your control, which has got your plus ampicillin. You've then got your agar, which has got your plus ampicillin. And then you've got your third plate, which is into control, which is your minus ampicillin. And then you've got with ampicillin present and your minus. So you need to go through and predict what the growth will be. Now the thing is, the virtual lab is going to be a little bit different to what we actually get, and that's okay, but we just want you guys to see what you can predict. Once you've predicted this, I want you to come and jump back in here and let us know, and there'll be a poll question waiting for your prediction. So I'm going to give you a minute to quickly jump into Canvas, type in your prediction. So all you want to do is put your lawn, your colony, and your no growth. Okay guys, so jumping back in, we'll miss the poll, and what we'll just get you guys to do is just raise your hand so we know that you're back here, so we can give you a next bit of instructions. Awesome, looks like you're all back, perfect. So just like I showed you all before, you're going to jump into the virtual lab and you're going to do steps up to number 4. So I'll just show you again on here. Just bear with me one moment. So on your virtual lab, you'll see at the top here it says step 1 to 11. I want you to get up to step 4. Once you're up to step 4, jump back in and we'll show you the next step live in the lab. Okay, guys, so you've got about 3 more minutes, and once you've done that, jump back in here with us. All right, guys, 1 more minute and then jumping back in. If you haven't finished, don't panic. You're going to have plenty of time to do more of the virtual lab later on. All right, guys, so we're going to be jumping back in here now. When you're here, can you please raise your hand so we can show you how to streak the plates? Okay, I'm going to move out of the way. Yeah, it's over there though. Hang on. Ah, I'm smart. All right, so Etienne's going to be streaking up the plate because he's better at it than I am. So bear with us as we change mics over.
Ooh, I feel like I'm on channel 7. Awesome. How are you all doing, guys? Don't answer that because I know you can't. That's fine. So what we're going to be doing today is we're just going to be streaking a plate. So it's a skill that's quite vital to microbiologists. Now, the reason we do this is because we are undergoing a process of dilution. So where did you put the ice bath? Yep. Awesome. Perfect. So the reason we dilute them is because although we can't see much in here, there's actually millions and millions in here. And if we try to get just one, it's just not going to happen. So what we're going to do first is going to crank the heat up. And as we did before, we're just going to pretty much sterilize our loop. See how it's going red there? Give it a couple of seconds to cool down. And because it's a liquid, we can afford to put it in when it's a little bit hot. And we should be able to hear a little when it goes in. Or not lucky today. Excellent. So the way we start is we start our primary dilution. So we're basically just painting. So here we go. I'm trying not to stab the gel here. Okay, and then we just gently just paint the surface. So although you can't necessarily see it, there's millions and millions of bacteria on here right now. So say we've got our millions. What we're going to do, we're going to sterilize it again. We want to get it hot, but not too hot that it maintains the heat, because if it maintains the heat, then we just melt through our agar. So we can touch it in the center just to cool it down. Excellent. Then we're going to do 3 or 4 just parallel lines. 1, 2, 3, 4. Excellent. So we've maybe diluted 1 to 50, 1 to 100. We can't say for sure, but now there's a lot less. There's still far too much, but a lot less. Now we're going to look again. Find my lines, there they are. We're going to do it again. So maybe a factor of 20, 50, something like that. Just going to paint that gently. Excellent. Then we're going to do maybe just 1 or 2 more. And this should hopefully get us down to maybe just colonies of 1 or 2. That's ideal. And then we go from there. So we're going to paint again, parallel lines. 1, 2, 3, 4. And then our last one, we'll do just 1 or 2 squiggly lines, and that will be our final dilution. If we've done it right, there should be next to none there. Excellent. So now that's finished, do our last sterilization, and this is ready to go in the incubator. Now, for the sake of time, I'll be doing the rest for you guys, but now we'll hand back to Kat, because that's the last.
Thank you. Thank you. Thank you. Sorry, guys. We're all trying to be COVID safe here. So now the next for you guys is we have done one of our plates, and if you were in our lab with us today, you would actually be doing all 4 plates with your partner, which is pretty cool. And then you're taken back to school to grow. Doesn't like my password. So you're taken back to grow, and then from there you would be observing your results and everything else. Okay, let me jump into here, one moment. That one back. So you can actually see what your next thing to do. So you've just watched us streak our plates. Now what you're going to do is jump in and do steps 5 to 8, and then we're going to be having your results. So jump back into your virtual lab. You're going to have 5 minutes to this in. So you've just witnessed us do that. And as I said, there are some things that are different in the virtual lab than we are doing in here. So 5 more minutes, jump back in. Hi guys, sorry to rush you, just a 1-minute call, and when you come back in, you'll see a poll waiting for you. All right guys, if you could jump back into Collaborate now. Oh, I can turn my Bunsen burner off now too, I don't need it. And you'll see you've got a poll waiting for you. It's waiting on a few more to answer. Two more to go. Okay. Looks like most of you done all right, and yes, half correct, that's pretty good. This is the wonderful world of science. You don't always get everything right. Okay. So we're going to get you to do your quiz to analyse your results at the end of this session because we're going to run out of time otherwise. And basically what we need to do when you do it is answer your questions in the virtual lab and then go to Canvas and answer the questions as well to analyse your results. So here is some of our results that we have, and I'm going to see if we can show you ours as well. So bear with me just one moment. So I'm just going to stop sharing. Yep, I'll just jump out of that view for a moment. So there's the pretty pictures. And hopefully this works. Can you get the lights, ET? Okay. So we've got our little UV illuminator here, and we've got, we'll put on plate number 1. You can see we've got some of our growth on there. Okay, we've got our plate number 2. This one's a bit hard to see, but you can start to see this one is particularly starting to glow. Then we have our plate number 3. And we're not always going to get the perfect results every time. And plate number 4. So you can see we didn't actually get any results on that one. And we got no growth happening there. So let's go back. We'll put our lights back on so we're not in the dark again. Okay. Whew, that was a bit weird. Okay, so I'm going to hide that. All right, so having a look here, you can see our results were different to what we've had before, but that's okay because it's always constantly changing. So we've got growth here on our first plate, and it's not glowing because it doesn't have the sugar present. We then have our second plate, which is glowing. we have our long growth on 3 and our no growth on our 4th one. So we're going to jump into our next prac and just quickly get as much done as we can. So we're going to digest our plasmid with E. coli R with a restriction enzyme, and we're going to put it into a gel. Now we've run out of time, so we're actually going to just have to show you the photo, but we'll show you the setup first. Why do you want to go through the process of this with our plasmids? Why do you think we want to go through? And so just typing in here, why do we want to digest our plasmid with our restriction enzyme? What's the purpose of that restriction enzyme? If you don't know, that's fine. Because this is a bit of an extra thing, but think about it. If we are cloning insulin, why would you want to digest it? That's a bit of a hint there. So if you're digesting the plasmid, which has your target DNA in it, your target gene, why do you want to digest it? Well done. Exactly right. We want to actually isolate that gene so we can pull it out. So we want to actually check the size bands so we can actually see that that particular isolated gene is in there. And we want to purify it so we can actually be like, yes, this is our insulin. Let's keep purifying it. Well done. Well done, Josh. So we're going to be doing our remote lab here. I'm going to stop sharing so you can actually see what I'm doing. Okay, so I'm going to jump around the other side. Actually, no, I'll just go here, it'll be fine. I'll push this back. Okay, so we've got our loading die and everything else. Okay, so we've got 8 minutes. Let's try and smash this out. So the first thing we need to do for our restriction, or our digestion, sorry, is we need to label our little Effendorf tubes. We're also going to be using our floater like we did last time. Alex, can you chuck me a floater from behind you? Thank you. So we've got our little floater because we're going to put it into the water bath. Luckily, we've got here's one we prepared earlier. So we're going to have one which our control, and this one we're not going to put a restriction enzyme with. We're going to have one which is going to have our E. coli R1, and then we're going to have our LD, which is our ladder. Pardon? No, we got our loading die here. I need my ladder, so it's all good. Okay, so the next thing, we're going to make sure that we have our tips being fresh each time. And we've got our sterile water here, beautiful. We're going to need 16 microliters first of our sterile water. Okay, grab our tip. So if you guys were here, we'd probably flip our experiment because you would be needing to be doing ET, this is a new one. Can you just chuck the right one in there? Sorry guys, got nothing to pipette in. Sorry guys.
So while we're just waiting for Kat, sometimes restriction digests are done before you do the bacterial transformation, and sometimes they're done after the bacterial transformation. So in this prac, now that we've seen that plate 2 shows the glowing bacteria, we know that it has been transformed. And then we take that and then we run it through a gel. Okay, we cut the plasmid with a restriction enzyme to see if we can isolate the insulin gene for further investigation. Sometimes you do a restriction digest before, so you can check what has run through the gel, and then you can grow it on the plates.
Thanks, Mabel. All right, so now we've got our product I can actually pipette. So we're going 16 microliters of our sterile water, and we're going to be putting it into our control. So it's just a tiny little dot. We are then going to be putting into that one our, where is it, that one there? Our P glow, so our plasmid. And I need to change my one. I need only 2 microliters of that one. So we'll turn that one around. Round and round it goes. Okay, so we're going to put 2 microliters of our plasmid in there. So it's tiny amounts. There we go. And then we do the same for our plasmid with E. coli. So you put in your 14 of your sterile water. You then put in 2 of your E. coli R1 and 2 of your plasmid. Then what we do is we simply chuck these into our water bath like we did before. And the cool thing is here's one we prepared earlier. So the next thing, after that's been in for about 25 minutes, we then chuck it into ice. Really important it goes from ice to hot to ice, because that way we know there's no more digestion occurring. So now we need to head over to our gel, and we are going to be placing our ladder in the center, and we're going to put in 13 microliters of each. So I need 5 and that one. So we're going to jump around the other side. We're going to show you the gel first. Can you get 3 tubes that are already? Just put 3 in there. Yeah, can you do that? Yeah. You probably need them. So I'm just going to move this chair out of the way. So over here is where we have our gel tank. And we've already put our buffer in here, and we've got our gel sitting in here, and we've got our wells. Now, what we do is we set it all up, put in our products, and then we put our gel cover on. And that runs for 45 minutes. I'm just conscious of the time, guys, and we don't want to keep you here longer than you need to. So Etienne's just grabbing our products that have been on the ice, and then we can get ready and pipette them in for you. Then after this, we're going to have a quick chat about gels and things like that. You right, ET? Yep. So sorry about the wait, guys. Okay, got the ladder as well? Perfect. Just chuck it in that tray. Thank you. All right, so we've got our ladder And we've got our products. Can I have the micropipette? I want 5 and 13. Okay, that's 13, so that's products. And 5. Beautiful, thank you. Okay, so now what we're going to do is put our ladder in the very center. And I saw this really cool thing online the other day that when they do this in labs, and I've actually put the video up for you, is that they pipette it onto a piece of parafilm so you don't get any of the product on the outside. I thought that was very clever, but I don't have time for that today. So I'm going to chuck it in here and going in on an angle and then slowly putting it in. Changing your tip there. And now I'm going to be putting in my products that we've had sitting on ice. So the first one is our control, which is that one. And we're going to put that into lane 1. What I mean by the lanes is what we have in our wells here. Actually, I'll put it in 2 today. So inject in the bottom. And then the last one is our one that's gone through. This is not a nice die today. The loading die is not happy. Okay. And then what we do from here is we put on our, on our tray, and then we turn it on. So it's all plugged in, ready to go. We select our program and we press start, and that runs for about 40 minutes or so. So what we're going to do is jump back over here, and as we don't have 40 minutes and a pretty one to show you today, which we apologise, I'm going to jump into our PowerPoint and we'll finish off our session. But we do have some pictures from our results from previously, so we'll show you them. Okay, so what we would get you to do is do a prediction. So if you can just quickly jump onto Canvas, do a prediction of what you would think you would see. So I'm going to give you 1 minute to quickly do that. So think about with your gel, one's been digested and one hasn't. So that's your clue. So jump into Canvas, you've got 1 minute, come back, and we'll continue with the next part. So 1 minute, guys. When you're back, guys, just raise your hand just so we know. Thank you. All right, guys, coming back, just raise your hand so we know that you're back. Awesome work. Sorry to rush you, but we know that you'd like a break before your next class. Okay, just waiting on a few more. So jumping back into Collaborate. And to make you feel a bit better, we don't have much more to get through, which is good. Okay, we got 80% of us? Did I turn on the right one? Oh no. Cool. Is everyone back?
5 more.
5 more. Jumping back in to collaborate. ET, can you check that I'm turned on because my computer's about to die? No, over here. Can you check that it's turned on? I was just switching through trying to make sure.
Good to go.
Cool, thank you. Okay, we got everyone. So just waiting on Charlie. Oh no, we've got Charlie. Chloe? Awesome. Okay guys, let's quickly jump through this. Thanks so much. So we're going to skip the virtual digest of using the restriction mapper. All the instructions are on your Canvas, so you can do it at a later time. So this basically tells you just putting that it's a circular, it's a plasmid, and you're selecting E. coli, and then you're just looking at what there actually is. The cool thing is you can put in whatever you want and learn a lot from your restriction enzymes as long as you've got your DNA. So we're going to have a look at our gel observations. Before we do that, we have a bit of a trick question here. So we've got our sheet of our agarose, which is your gel, and we're putting in our digested DNA up here, as you saw. We can put our current over, but why do I put it here and not down where the positive end is? Can anyone tell me why? Your clue is to do with your DNA. Why do I inject at the negative end and not the positive end? With the charge.
Your clue. I cut you off, sorry.
Okay, so your clue is to do with the charge of your DNA. Look, what are our answers? Well done. DNA is negative. Perfect. So therefore, it will flow towards the positive end. And size does matter depending on how big your band is. If you are small, you're going to be able to get through that agarose gel a lot faster than a larger piece. So here's a bit of a picture to give you a hint. If you're an elephant-sized one, you're going to take a lot longer to get through than your little mouse. And here's a bit of a skill for you guys. I'm just going to randomly say C. Who can tell me what C is? What is its base pair length? So how many base pairs are on C? So this is really important to be able to read yours. So yeah, you're looking at about 700. It's just a little bit below it, but well done. It's really hard because with your gels, you have to give a bit of an estimation. Great work, guys. So here is our gel that we've previously run. So our gel's still running over there. So lane 2, you've got 1 band here. Lane 3, you've got 2 bands. So what is this telling us? What's it telling us? Well, we've got our E. coli here, so we know this one has been digested. This one hasn't. The really cool thing with plasmids, because they're a circular piece of DNA, they can actually turn and supercoil, which actually can give sometimes 2 bands, which is pretty cool. The last thing is once you've done all your questions, you've got your conclusion, and we want you to write basically how these 2 practicals, well, from your knowledge, to produce insulin, and why is bacteria used to produce insulin. So this is just some questions to get you starting to think about your conclusion. So let's recap, guys. Hopefully by the end of this session, you can start to see why we use restriction enzymes for digest. Sorry, we had to rush it a little bit, and that you're able to observe and do your own virtual lab with a bacterial transformation. And lastly, to evaluate the growth on the plates, to work out who has been transformed and who has not. Thank you so much for joining us today. There is a post-program survey that we'd love for you to answer once you finish up. The other thing is you've also got some post work to go through, some more exam questions to practice. There also is an additional module, which is, I think, number 7, which is about PCR. And so there's some questions in there and things like that. So good luck with your exams. Thank you so much. I've tried to make this as fast as possible. I'm going to hand over to your teacher. If she's got any questions or if you guys want to ask anything, please stick around. Otherwise, thank you so much. I'm going to stop sharing. So I'm going to hand over to your teacher now, and she is going to give your instructions.
Hi guys, just you can leave now. We've got a class tomorrow morning and we'll discuss some things there. So sorry, we've only got about 5 minutes.
No, that's fine.
Yeah, if they can, you can all just jump off and we'll go through this tomorrow morning.
Sorry it was rushed at the end there.
Thanks for today.
No worries, absolute pleasure. Sorry it was rushed at the end there.
Yeah, I just think maybe some of them struggled a bit with the context at the start.
Yeah, that's all good.
Yeah, because they didn't know what P-glow was in context. So yeah, and I'd got another class on Thursday. Just a preference for me. The micropipetting, is not that important.
Cool.
In context of if they're doing it, it's important, but I probably would have rather them spend a bit more time on the gel electrophoresis.
Yeah, no, that's fine. Thank you for the feedback.
Just a little bit of feedback. So I think, yeah, it's really, because I've actually got 2 P-glow kits sitting